All right, so I'm going to make one kind of longer mitosis presentation for lab, and we're going to be going over the cell cycle. We're going to talk about some things about DNA and then, of course, go over mitosis. Most of this stuff is not going to be microscope work except for identifying the different stages of mitosis. So I'm going to open up our slideshow and we're going to go through mitosis and talk about the different stages. So the first thing that we need to know about cells is that most of the cell's time is going to be spent just in a state of normal cell function called interphase. So where it says G1 and S phase and G2, these are phases where the cell is going through its normal function, especially in G1. The cells aren't dividing very often, actually. And so they're going to be in this G1 phase for most of the time the cell's alive. When the cell decides it needs to divide, it's going to go into the S phase where it replicates its DNA. And then it's going to go into the G2 phase where it prepares to go through mitosis. Remember, mitosis, hopefully you remember this from biology class, but it's broken into 4 steps called prophase, metaphase, anaphase, and telophase. And we go from having one cell to having 2 identical cells. So before we get into mitosis, we need to kind of look at DNA just a little bit so that you can remember some of the things that we talked about at the beginning of the semester. So remember, DNA is made out of nucleotides. The nucleotides are built of 3 parts. They have a sugar and a phosphate group that make up the ladder, kind of the sides of the ladder of the DNA. And then they have a base, which is our genetic code. So these 3 molecules combined are called a nucleotide. And so when we put nucleotides together, we build our DNA. So see here, we've got the sugar and the phosphate group of one nucleotide here with its base. And then we have a second one right here with its base. And the phosphate and sugar make up the sides of the ladder. And then these bases make up the genetic code. They make up the steps of the ladder. And remember, we have 4 bases: thymine, adenine, cytosine, and guanine. You also need to remember that thymine always bonds to adenine and cytosine always bonds to guanine. There'll be questions about this on the test, so make sure that you know that. Okay, so we put all the nucleotides together and we make up this ladder of DNA. Now remember, the ladder is a twisted ladder called the double helix, and there could be a question about that. So when we look at this, see, we have all of these phosphate and sugars making up the side of the ladder, and then all of the bases making up the steps of the ladder. So when DNA needs to replicate itself, when it needs to make a copy to undergo mitosis, we can unzip the DNA and then read the A's, T's, C's, and G's and make a copy. See, we have one copy here, but then we are splitting the DNA open and we're making 2 identical copies. See, we have A and T and T, G, C, G, C. We have 2 identical copies. One of them is going to go to one new cell. The other copy is going to go to the second new cell. Now, in order to do this, we have to copy all of our DNA. So what's interesting about this is our chromosomes are only X's when the chromosomes are copied. So see, if you look at the left-hand side of the X, it is a mirror image of the right side. Most of the time, chromosomes do not look like X's. They look like capital I's. And so we only have one copy of DNA here. the DNA copies itself and makes an X. And then during the cell division, during mitosis, one set of the X chromosome goes to one cell, and then the other one goes to the second cell. So that's how we duplicate our DNA. The DNA is held together with the X, the two copies, in this little center part, this little center connector called the centromere. The two copies of DNA or chromosome is called sister chromatids. So they're going to break apart and one's going to go to each cell. And the two new cells that are made are called daughter cells. Pretty cool. Okay, so we need to know these phases of mitosis for the lab test. And so we have prophase, metaphase, anaphase, and telophase. And if we go to the next slide, we can see that the cells in interphase are kind of just doing their normal cell function. They have a nucleus. You don't really see any DNA because the chromosomes are unraveled into what's called chromatin or chromatin. When the cell starts to get ready to divide, the first stage is prophase. And in prophase, what happens in prophase is the nucleus of the cell breaks down. We form all the copies of the chromosomes, and the chromosomes thicken up so that we can see them, and then they are just visible. So you can see all these chromosomes kind of in the middle of this real cell. After prophase comes metaphase. The chromosomes line up at the center of the cell, and they are held in place by these spindle fibers, these proteins that are grabbing onto the chromosomes. Now, one copy is going to line up on one side, the other copy is going to line up on the other side. During anaphase, these chromosomes are going to split, and one copy is going to move to one end of the cell, and the other copy is going to move to the other side of the cell. The movement of the chromosomes is anaphase. Anaphase stops when the chromosomes reach the ends of the cell. We enter telophase. In telophase, the nucleus is going to form around those chromosomes again, and then the cell is going to divide. So we get what's called a cleavage furrow. It's kind of this pinching off, and we pinch the two cells apart, and we have what's called cytokinesis, where the cell completely divides into two. And then the cells go back into interphase. Kind of cool. Here is just some more examples of what to expect when you see this. So here is just interphase with the cell doing its normal function. In prophase, we break down the nucleus and the chromosomes copy themselves and form. So this is kind of what that's showing here. It says, don't worry about pro-metaphase. Just think of this as prophase. So in prophase, we lose our nucleus. The chromosomes have copied themselves. Then during metaphase, they line up and are attached by these spindle fibers. The spindle fibers then pull the chromosomes apart. And in anaphase, one copy goes to one end of the cell. The other copy goes to the other end of the cell. And the movement of the chromosomes is part of anaphase. Then we get into telophase, where the chromosomes stop moving. A nucleus can form, and the cell then starts to form a cleavage furrow, and it divides through cytokinesis. And then we end up with two identical cells. So what you have to do for the microscope work is you have to be able to look at these whitefish blastula. Whitefish, we eat whitefish, like fish sticks are made out of whitefish. So these are all just like whitefish eggs, basically. Or whitefish, a blastula is like an early embryo, like a very early embryo. So we're looking at whitefish embryo, and you have to just kind of look at each cell and identify what phase they're in. So like this phase is just interphase. It's not even going through cell division. And then like this guy is in anaphase. It looks like those chromosomes are moving. So here on the last slide are just kind of the phases of what you should look for. So this first picture is just interphase, where the cell's doing its normal stuff. Then the nucleus is going to darken up as those chromosomes form during prophase. So if you see a really dark nucleus, or a nucleus that's breaking down, maybe it's kind of looks like blobs, that's prophase. Metaphase, you'll definitely see like a line of chromosomes forming as the chromosomes line up in the center of the cell. Then in anaphase, you'll see them getting pulled apart. You don't see the movement, of course, but you'll see them getting pulled apart. And then finally, in telophase, you'll start seeing two cells with the cleavage furrow, and the chromosomes are now in each cell, and they've been pulled apart. You might even see a nucleus forming. All right, everyone. So that's it. I mean, there was not a lot in lab to do. So what you have to do in lab for most of the week, make sure you go over this, but go back and review your blood, your lymphatics, do the blood types, make sure you know your skin really well, because when I get back next week, we are having our test on the block day. The lab test will be on the block day. We'll probably start some new lecture stuff on the digestive system. We'll plan on having our lecture test the following week. Then we have spring break coming up. And then, we're on the home stretch. So I think we're on track. We'll have 3 tests in lecture done, 2 tests in lab. That is technically on track. We just got to keep rolling. So good job. I hope you're having a good week, by the way. Make good choices. I will see all your smiling faces on, I think it's March 3rd is the Monday. So until then, take care, and I will see you soon.